Hello everyone. We will start in 2 minutes. Let all of the participants join and then we will start. Good evening, all of you. My name is Ubaid Tariq, and today is the second session of this problem solving NPTEL session. In this session, I will solve the problems of week 2 assignment of cell culture technologies. This course. So we will start with the questions. So first question is one of the major roles of carbon dioxide in cell culture is to. You can unmute yourself if you want to answer, or you can write in the chat box if you know the answer. Anyone from group members? One of the major roles of carbon dioxide in cell culture is to. The answers are imbalance pH, balance surface tension, imbalance surface tension. Yes, the answer is majority of you have given the correct answer. The answer is balance pH. So CO2, the only role of CO2 is to balance the pH of the media which is used in the cell culture. Can anyone tell me how much percentage of COH is used in cell culture? How much percentage of CO2 is used? The percentage of CO2, which actually depends on the media, the concentration of bicarbonates present in the cell culture media, and in most of the medias this bicarbonate concentration is such that the 5% CO2 is required to balance this reaction so that the carbonic acid and bicarbonate buffer is formed. So here the answer is the CO2 is used to balance the pH. As you know that in our body we assimilate only oxygen. CO2 is not the metabolic requirement of our cells, human cells or cells of any mammal. They may be required at some points, but the metabolic requirement, CO2 does not fulfill any metabolic requirements of the body. So cell culture, we also do not need to add the CO2, but to maintain the pH, we add CO2. So usually 5% of CO2 is added. For example, if this is the cell culture flask, in the cell culture flask, in the bottom of cell culture flask there are cells here, and above the cell these cells there is a liquid phase which is the cell culture media, and above is a gaseous phase where there is the 5% carbon dioxide. This 5% carbon dioxide, it gets dissolved into the liquid phase of the cell culture media, and then it gets converted into carbonic acid. And there are also bicarbonate buffers present in this liquid phase in the media. Those bicarbonate buffers, they can dissociate into protons and carbonate ions, and this carbonic acid and bicarbonate, they balance, and they will lead to the formation of buffer system, bicarbonate buffer system, which will ultimately resist the change in pH. As you know that buffers have the property that they resist the change in pH. They do not allow the to change in pH. They resist in some. So I will explain more things. Now you may have the question why we need these buffers uh, in the cell culture media. So there are buffer systems to resist the pH, but there are also the indicators. Phenol red is an indicator which is used in the cell culture to help us to visualize the change if there are any change in the pH. For example, uh, if in any case there is a contamination in the cell culture media, so you have to visualize the flask, you have to visualize the cells in the microscope to see if there are any contaminations or not. But as the phenol red indicator is present in the cell culture media, so if the phenol red indicator turns yellow, there may be the chances that there is the contamination. If you see your flask media has turned yellow, then you must be curious that maybe there is the contamination, and then you can check in the microscope how your cells are looking. And this phenol system, this phenol red above this indicator, it also gives us the idea when cells need when we need to change the media of the cells. For example, if the color of the media is little bit yellowish, not that much, like this color, so you can wait for 24 to 48 hours. After that, you can change the pH. And if the color is this extreme yellow, so the culture media is very acidic. There are more metabolites which are secreted by the cell, acidic metabolites which are in the cell culture media. So we need to change it that day only, or we also need to check for the contamination. In case of contamination of if the media is very acidic, so in that case, the media looks like this left side one, the pH 6.5. So in that case, you need to change it on the same day. But here, in if the pH is 7, the media looks like this. So you can wait for 20 to 48 hours to change the media. But if the media is like this, you can leave the this. You can leave the cells because the media is okay. So it gives the visualization without looking the cells in a microscope. You can by looking at the media, you can say when we need to check, when we need to change the media, or if there is any contamination, you can have an idea of that. If the this color of the media is more this reddish or this pinkish color, so then you need to check the CO2 supply. So pH must have increased to 7.8. So in that case, you need to check the CO2 supply if CO2 is going in the cell culture media. So when CO2 goes in the cell culture media, it actually reacts with the H2O present there and forms the carbonic acid. If CO2 supply is not there, the carbonic acid will not be formed. So there will be imbalance between in the bicarbonate buffer, and the pH will be more basic. As I already said in the previous lecture, that in case of various types of contaminations, you have to if there is a change in color of your media, so there may be chances of contaminations in your cell culture plates or cell culture flasks. What are the different types of contaminations? I will explain in this slide. So first picture, this bacterial shows the bacterial contamination. You can see these are the cells, mammalian cells, and these particle type cylindrical entities, they are the bacteria. And in the when you see them in the bright field microscope, you can see them easily at 10x or 20x. So you can also see them moving around in the cell culture media. So in that case, you have to discard your cells because this it is very difficult to remove or kill these bacteria. So you have to discard your cells. Another type of contamination is fungal contamination, in which fungus grows in the cell culture media. And in the microscope, when you see the cells, you see like this mesh type structure. This mesh type structure is a fungus. There is a fungal contamination. Then there is a fungal contamination in your cell culture plates. Another type of contamination is yeast contamination. So if you see this type of bubbles or these type of particles in your cell culture media, it may be the yeast contamination, but sometimes this yeast contamination it may be confused with the dead cells. When these cells die, these are the adherent cells. When these cells die, they get detached from the cell culture plate, and they also get circular like this. But the difference between this yeast and this dead cells is that in the yeast, as you know, that yeast it divides by budding. So you can see like bud-like structures here in the yeast contamination. If there are these bud-like structures, there is then definitely there is the yeast contamination in your cell culture. Another important contamination is the cross contamination. If someone is working on two cells, for example, hepatocytes and cardiomyocytes, and somehow he touched the pipette of hepatocytes in the flask of cardiomyocytes. So there may be the cross contamination. Some of the hepatocytes will go in the cardiomyocyte flask, and they will grow. And it is very difficult to detect this cross contamination. If the morphology of both the cells, for example, smooth cardiomyocyte have the same morphology. If the morphology of cells is same, then it is very difficult to detect under the microscope that your flask has a cross contamination. So you have to be vigilant. If you are working with more than two cells, you have to work separately for both the cells. But in some types of cells, if there is a clear difference in the morphology in the microscope, if you visualize the cells very keenly, you can find that there is a difference in morphology, and there may be two types of cells growing in the same cell culture plate. And you have to be vigilant, because these cross contamination will give wrong results, and you will not be knowing that there are two cells, not the one cell. Question number second is, entry and exit of a lab should be clearly defined and not obstructed to ensure. What should be the answer? A part is safe exit in case of emergency evacuation, safe and entry of external air in the lab, aesthetic appeal of the lab, nobody can enter the lab. What should be the answer? Deepa, you are not able to see the question.
A. Sir, answer is A. Safe exit in case of emergency.
Yes, safe exit in case of emergency. And the answer B, it is very much incorrect, because in cell culture it is ensure that the external air, it does not enter the cell culture lab. So different that door that air which supplies the high pressure air, they are placed outside the door so that external air will not enter the cell culture. So the answer is safe exit and entry in case of evacuation. For example, if any mishap happens in the cell culture, maybe the bursting of CO2 cylinder, maybe some fire. So in that case, there should be safe exit for the evacuation. Is my slide visible? Someone was saying that question is not visible.
Yes, sir.
So in this slide, I have shown that what are some personal protective equipments as we are talking about entering the cell culture lab. What are some PPEs, personal protective equipments that one should wear before going to the cell culture lab or before working in the cell culture lab? Because uh, we have to ensure the cleanness of the cell culture lab. Also, we have to ensure the safety as of our cells as we are working with the cells and different. So we have to, or different molecules are there. So different chemicals are also there. So we have to work for the safety of ourselves also. So these are the following things that you should take care, like wear your head cap so that the hair fall, which fall, they should not fall in the working area. Wear the mouth mask, face mask you have to wear the lab coat, gloves. You have to change your shoes so that the outside shoes, uh, they may be dirty or they may have some contamination, so cell culture shoes should be separate, or you can wear the shoe cover so that your shoes will be covered, and you have to wear the gloves and lab coat to prevent your clothes to get dirty or to get some contamination which may harm you. And before entering the lab, you have to clean your hands with sanitizer, or you have to wash your hands with the soap. Then you have to enter the lab. You have to enter some common area between the cell culture lab and between your normal lab. So in that area, you can these things are placed. These gloves, lab coats, masks. These are placed. Different shoes or shoe covers, or sleepers. They are placed so that you can wear that. Third question is aseptic techniques are employed in the cell culture lab. Answers are to contaminate cultures, to prevent contamination of cultures, to remove dust for the aesthetic appeal of the lab. So answer is B, to prevent the contamination in the cell culture lab. As I showed you in the earlier slide, that the contamination is the very serious problem in cell cultures. Sometimes you work for 10 days, 15 days, and you are growing cells. You are increasing the number of cells. Maybe in your experiment you need the large number of cells, large number of flasks. At the 15th day, or your cells, if you not follow these aseptic techniques, if your cells will be contaminated, all the 15 days are lost, all your cells are lost, your flasks resources all are lost. So you have to follow these aseptic techniques very strictly so that to prevent the contamination in the cell culture. So in the cell culture lab, there is a cell culture hood, which may be the basal 3 or basal 2. Usually in cell culture labs, basal 2 hoods are there. So all the procedures, like if you are giving the treatment to the cells, or you are adding the media, you are removing the media, you are splitting the cells, whatever you are doing in the cell culture plate, all the procedures should be done inside this laminar airflow. And also media, your flasks, they should only be opened in this cell in this laminar airflow. So what is the working principle of this laminar airflow? It contains the HEPA filters, high particulate efficiency filters, and air is circulated between these HEPA filters, and external air does not go into your this working area where you have placed your flasks, your media, and all other things that with which you are working in the cell culture lab. So the air from the out, in some cases the outside air will also go, but here is the vent. It can directly go in the bottom of this laminar airflow, and it will not go into your the working area of your laminar hood. And after that, it is filtered through these HEPA filters, and after filtering through HEPA filters, it is again filtered. Maybe in some cases there are two layers of HEPA filters, and after that, it is supplied to the laminar, this working place of the laminar. And HEPA filters, the size is usually 0.2 to 0.3 micrometer. So all the particles, like your bacteria has a size of 2 to 3 micron, virus also has a 0.3 micron, all the dust particles or spores, they have the 0.3 micron size. They all are removed uh, using these HEPA filters. So you get the clean air when it is passed through HEPA filters. So the air which is, it is a clean air, it does not contain any bacteria, any virus, any particles, any spores. So so all the procedures should be done here so that your cells will not should not be will not be contaminated in this laminar airflow. So this is the front view of the laminar airflow, where there are the this is the working area, and these are the some things, some equipments or some reagents that you require when you work in the cell culture. This is the 70% ethanol spray. When everything, every item when you enter them into the this cell culture hood, you have to spray the 70% ethanol. And when you also place your hands for when you are working in the cell culture lab, you have to wear the gloves, and you also have to spray 70% ethanol. And here is the liquid waste. If you change the media of cell culture flask, so liquid waste is placed in the left side. So you have to divide this in the different portions. For example, this left side, sorry. For example, this left side area, it contains all the waste items. For example, waste liquid or any some polythenes or covers which on in which there are these cells or falcons. So that have to be, you have to place in the one side, left side. For example, all the reagents or pipettes you can place on the right side, and between that you can place the cell culture flask where you can work or where you can open. So you have to, so that you can prevent the contamination. So you have to separately divide the portions of the cell culture, the cell culture whole. Fourth question is, which of these is used in cell culture lab to store cell line vials for long term? Liquid oxygen, liquid carbon dioxide, liquid nitrogen, liquid hydrogen. Answer is, yeah, most of you are answering correctly that cells are usually stored in a liquid nitrogen. So these are the liquid nitrogen tanks. In these tanks, liquid nitrogen is filled, and these are the stands or racks in which you place these cryo boxes. And in these cryo boxes, there are the vials. They are called cryo vials, and these racks will be dipped in the liquid nitrogen. And when you have to take the cells out from this liquid nitrogen, so you have to remove these racks, as there is a handle above there. You have to remove these racks and open the box, these cryo boxes, and you can use your samples, cell samples, or cells that you have stored. So why we need to store the cells? For example, if you are working with the cells, you have done your experiment for 10 to 15 days. After that, you are planning your experiment for after some time. for example 10 days, 15 days, 1 month, but you cannot keep your cells going, running in the cell culture as they are growing. So you have to store them. So storing the cells as they are live, so you have to freeze them so that all the metabolic activities of the cells are froze, all halted, and also there should not be any damage to the cell, these cells. So these the cells, after this cryopreservation of, or after the storage in this liquid nitrogen, you should be able to revive, or you should be able to reuse them when you thaw them, or when you take them from this liquid nitrogen, maybe after a year or 2 years. So you should be able to use them. So in that case, so proper arrangements, or proper, you have to freeze them very carefully. So any one of you know about cryoprotectants? What are cryoprotectants? Anyone? So cryoprotectants are the chemicals or substances that protect the living things or living cells when they are in the frozen state. For example, in the cell culture, usually glycerol or DMSO, dimethyl sulfoxide, these two are used as a cryoprotectants. So how they protect the cells? If you freeze the cells directly place the cells in this liquid nitrogen, and its temperature of this liquid nitrogen is minus 196 degrees centigrade. Uh, or sometimes these, if the cells need to be stored for a shorter time, you can also store at minus 80 degree deep freezer, minus 80 degree freezer. But in both the cases, the as you know that in our cells, the cytoplasm, or the which is the aqueous phase, and it gets frozen when it freezes, and it after freezing it, the ice crystals may be formed in the cells, and these ice crystals, they damage the cells. And uh, and outside also, in the media, ice crystals get formed, and these ice crystals, they are very pricky, and they damage the cells. And after that, cells are very they will die in the storage conditions, and we will not be able to revive the cells after some time, after maybe if we revive after one year or 2 years. So these cryoprotectants, what they will do, for example, DMSO, when DMSO is used, usually 5% or 10% DMSO is used, because DMSO is toxic up to cells after 10% concentration. So this DMSO, it abruptly diffuses inside the cell, and because of this DMSO, these ice crystals will not be formed in the cell. So it protects the cells at the time of cryo. Cryo means freezing. So it protects the cells at time of freezing. So ice crystals will not be formed and cells will not be damaged. So when you after 1 or 2 years, when you thaw the cells, so you have to remove this media which contains DMSO, and you have to put the fresh media which contains the only FBS and uh, this defined media. So I hope this is clear. Question number 5 is HEPA stands for high efficiency particulate filter, high efficiency pyramid filter, health and emergency patient accessibility, high efficiency primary flow. So the answer is A, high efficiency particulate filter. As I already explained you in the previous slide, these MR hoods, they contain these HEPA filters, and air is passed through these HEPA filters. And these HEPA filters have the size less than 0.2 micrometer, and all majority of the viruses, bacteria, the spores, they have the size more than 0.3 micrometer. So all these, they will be blocked from entering into this working space of the cell culture. So they will prevent the contamination in the cell culture. So they are high efficiency particulate filters, the full form of HEPA. So here in this picture, it is also shown that this is a contaminated air, which it may contain the particles, dust particles, viruses. Uh, but when it is passed to the HEPA filters, the clean filtered air is supplied in the working area. Which one of the following is not safe to be autoclaved? The answer is stainless steel, high density polyethylene, surgical equipments, glass. So most of you have answered correctly. Answer is B, high density polyethylene. All the other items like stainless steel, surgical equipments, they are usually made up of steel, and glass, they are all autoclavable. But this polyethylene is a polymer, so it's not autoclavable. It gets destroyed or melt. It melts in the autoclave. So can anyone tell me why do we use autoclave? In case of cell culture, we frequently use autoclave. So autoclave is just like the pressure cooker. So there are four components of autoclave, four things that are important when you autoclave the item. One is the steam, another is the temperature, another is pressure and time. So there should be the some particular amount of all of these things to attain the sterility, so that the spores, microbes, viruses. Yes, for sterilization purpose. Someone has answered that. For sterilization purpose, we use the autoclave. So this is the out line of the autoclave. It contains the heating element, which heats the water which is present below this perforated tray or this mesh. And this, when this water boils, the steam is generated, and this steam, and here in the top, there is a pressure gauge, safety valve, and this lid. And this lid is placed here. It is tightly placed here, and all the items that you need autoclave are placed here. But as you know that the temperature of the boiling temperature of water, what is the boiling temperature of water? 100 degrees centigrade. So if we boil the water without the high pressure, without the if the water is boiled at the open place, so the vapors which are generated from the water or water itself, they will be only heated up to 100 degrees centigrade. Above that, they cannot be heated. But in case of autoclave, as this lid is closed, so pressure is also generated. So because of this pressure, the temperature can be elevated inside this autoclave. So, so there is also the correlation of temperature and the time for which autoclaving should be done, so that all the microbes, spores, viruses, they will die. So if you directly boil your equipments that you want to use in the autoclave, maybe surgicals, blades, your flasks or tips that you want to use, if you boil them in the water, so you have to boil them for the 90 minutes at, because water only boils at 100 degree, so you have to boil them for 190 minutes to ensure that all the microbes or all the spores or all the viral load is removed from these equipments, and they are sterile now. Because the pressure is not built up, so the temperature remains 100 degree only. But if this lid is closed, and the pressure, when the pressure reaches to 5 pounds per square inch, so when it reaches to 5 lbs, the temperature can reach to 110 degree at that time because of the pressure. The temperature of the steam will reach to 110 degree, and at 110 degree only 60 minutes uh, time is needed to sterilize or autoclave the things. If the temperature is 10 pounds per square inch, so the only temperature will reach 116, only 30 to 40 minutes will require. And usually in autoclave, there is a 15 pounds pressure, and we autoclave it for 15 to 20 minutes, and the temperature at the time will reach 121 degrees centigrade. And here we only need to autoclave for 10 to 5 minutes. And usually in the autoclaves that are present in the labs, this 121 degrees centigrade pressure temperature is maintained at 15 pounds per square inch, and we do autoclaving for 10, 15 to 20 minutes. In some cases, if you want to do it more quickly, so you can increase the pressure. So this pressure gauge or this valve is adjusted such that it can build the more pressure. And if it's adjusted at 15 pounds, that when the pressure reaches 15 pounds, it will lose some pressure, so that the pressure is maintained at 15 pounds. Temperature will only be risen up to 121. But if we adjust this gauge or the things that are provided here, if we adjust the limit of 20 pounds per square inch, then in that case temperature will automatically will reach 126. You don't have to adjust that, so you have to adjust the pressure only. So this case, if the pressure is 30 pounds per square inch, in that case the temperature will reach 135 degrees centigrade, and only 3 minutes of autoclaving is needed. So temperature is inversely proportional to the time for which autoclaving is done. And what's the importance of this steam? Why we do not use directly the water uh, to autoclave? Because the steam has the more penetration power in the bacteria viruses. It penetrates more quickly and easily, and it will lead to efficient autoclaving or more quick autoclaving or more quick sterilization. So next question is, dash properties of cells are required to relate the cultured cells to functional cells in the tissue for which they are derived, from which they are derived. So what are the properties that are required so that the cultured cells are functional? Dedifferentiation, apoptosis, differentiation, aneuploid or aneuploid. Some of you answered B, apoptosis. Why we need to apoptose the cells? In the last this session, I have explained about the differentiation, redifferentiation, differentiation. If someone have joined that session also, they can recapitulate those things also. So the answer, this very few members have given this answer. The answer is C, differentiation. I will explain how in the next slide. So usually the cells which have the pluripotent or totipotent nature, they are less specialized. So they have less specialized function also. Their function is only to make the different types of cells. But in the cell cultures are grown in such a condition. Here the question was, to, uh, dash properties of cells are required to relate the cultured cells to functional cells in the tissue. So for the to making the functional or specific cells, differentiation is the answer. For example, these stem cells, they have the property that they can differentiate into any type of cell. For example, transcription factors or different molecule transcription factors are given in such a way, they are provided to these stem cells. They from the osteoclasts, and from these osteoclasts bones are formed. If different transcription factors can be given in different cases to specifically differentiate these stem cells into the different types of cells, for example neurons, enterocytes, hepatocytes, and cardiomyocytes. After this differentiation, they do some specific function. So they are now they are functionally specific. They have some function. For example, cardiomyocytes are in the heart. They have the pumping ability. These osteoclasts, they are in the bone, and they have the mineralization. Hepatocytes are in the liver. They have their own function. Intestine cells, they assimilate these food and the drugs. They take them in the blood. Brain neurons have also their own function. So after the differentiation process, the cells become specific to any function. So differentiation makes them more functional. Next question is, using a pH meter, you find the pH of an unknown solution to be 8. How would you describe this solution? This is the very much basic question. So what is the answer? Strong base. If the pH is 8, it's a strongly basic, weakly acidic, strong base. So everyone of you should know this. So if the pH is 8, the solution is weakly basic. So here in this picture I have shown the pH scale between 6.5 to 7.5, we call it, or around 7 we call it the neutral pH, and the range, whenever the far you go from this 7, for example, if you go from 7, you go from the left side. So if the pH is 1, 2, 3, or 4, the acidity increases, so these are more acidic, and which are close to this neutral pH 6 to 7 or 5, they are weakly acidic. And here in the right side also, the if the pH is if the difference is more than 7, for example, if it is 11, 12, 13, it is more basic, and the pH which is close to this neutral pH, like 7, 8, 9, it is the weakly basic solution. Another question is, dash light is used to sterilize the air in the laminar flow hood between uses. So the answer is UV light. So why it is necessary to use the UV between uses? Between uses means if the one person is doing some procedure in the cell culture, and after that another person wants to use the cell culture hood, so between that the UV is given to the cell culture hood, or for 15 to 20 minutes. So the reason is, when the one person works, maybe the media is spilled in the floor of this laminar air flow, laminar air hood, there are any contaminated pipettes, or which are contaminated with the cells. It may not be the bacterial or viral contamination, maybe the contamination of the cells also, or maybe in some cases bacterial or viral contamination also. So you have to give the UV to cell culture hoods for 15 to 20 minutes between every use. And in the morning, when the first person starts the cell culture, the first person also have to give the UV to the cell culture hood, and also the UV should be given once in a day, in the morning, to the whole cell culture room, to ensure that this all the cell culture room is sterilized. But in the laminar airflow, it is more strictly followed, and between every uses you have to give the UV. So what is the rationale of giving the UV light? So UV, the wavelength of the UV, it ranges from 100 to 400, but the effective, which is majorly effective, is the UVC radiations. These UVC radiations, they have the higher penetration power as compared to this vacuum UV, or as compared to this UVB and UVA lights. And no, sorry, this UV range is between 200 to 400. Sorry, I said from 100. It is between 200 to 400. And from this 200 to 280, the it's a UVC, and 280 to 315 is a UVB, and 315 to 400 is a UVA. And the UVC, it has a lower wavelength as compared to other UV lights, and because of the lower wavelength, it has the high energy, as you know that the correlation. So when there is a lower wavelength, there is a higher energy, so that they have the more penetration power as compared to UVB and UVA. So they penetrate the membrane of the bacteria, virus, spores, and they what this UV light do, it will lead to the it will damage the DNA present in the bacteria, virus, or other fungus, yeast, or other cells which are present in the cell culture hold. So this UV light, this will lead to the damage to the DNA present in the microbes, and after that these microbes will not be able to do transcription, translation, so the proteins will not be formed, and also microbes will not be able to divide, when and ultimately these microbes will die, and they, they will not now contaminate the cell culture. And usually this UV light, it forms the thymidine dimers. These thymidines, they dimerize, and these bonds between these two strands, they are broken down, and the, it will ultimately damage the DNA. Another question is, a major goal of embryonic stem cell biology is to develop protocols that enable lineage specific control of dash division, differentiation, survival, all of them. So in embryonic stem cell biology, or you can say stem cell biology, what type of protocols should be developed to enable a lineage specific control of, most of you have answered all of them. Oh, yes, that is the correct answer. So the protocol should be developed so that to control the division, differentiation, and survival of the cells in these embryonic stem cells. So as I already said in the earlier slide, so these embryonic stem cells, they have the totipotent or pluripotent potential. So when they have the potential of developing into any type of cell, so if we control the, if we provide some, for example, in case of differentiation, if we know, if we know the protocol of developing cardiomyocytes, protocol of developing hepatocytes, smooth muscle cells, epithelial cells, so we can control the process of differentiation. For example, uh, actually these embryonic in our body at the time of embryogenesis, these things happen in our body. But if we develop the protocols, or if we know the protocols of these, so we can use different transcription factors. For example, here I will go to this slide. So if we specifically want to make the cardiac cells or cardiomyocytes, and if we know the transcription factors which are required so that these stem cells will make these cardiomyocytes, so we can take stem cells, we can add these transcription factors, and we will get cardiomyocytes. So any type of cell we can get in days using the transcription factors. So this will be very helpful in case of tissue engineering, so that you can make any type of tissue using these stem cells in vitro conditions. So in vitro conditions, you have the stem cells, you will give the transcription factors, and these transcription factors will lead to the formation of different cell types, different functional cell types. And these stem cells, they also have the limited survival. They they do not survive for different passage in the in vitro conditions. If you control, if we develop some procedures or protocols that will help to enhance the survival, it will be also beneficial for the stem cell biology. And another thing is the division. If we control the division of the stem cells, and after differentiation, the division of the functional cells, or differentiated cells which are made from the stem cells, so it will be beneficial to specifically control the lineage. So here in this example, like progenitor cells have the ability to develop specific lineage of cells, for example myeloid lineage or erythroid lineage. So so to from these progenitor cells or stem cells, different transcription factors are needed, which are lineage specific transcription factors. For example, if we want to make the myeloid lineage, so different lineage specific myeloid transcription factors are required, and which will lead to the transcription of some signaling molecules or some proteins. After the transcription of the signaling molecules, the cells will be changed into this myeloid lineage. And if and in some cases, if the transcription factors are provided for this erythroid lineage, so these will lead to the formation of different signaling molecules, which will ultimately lead to the different lineage of cells, erythroid lineage of cells. So we can control the differentiation of the cells. So this is all about the questions, 10 questions which are provided in the assignment. So this is my YouTube channel where I will upload all these videos of this live session. If you miss some part, you can go to this channel and you can have a look of these videos. I have also uploaded other videos, previous videos and other videos that may be helpful for you. So thank you for joining this session. If you have any more questions, I will be happy to answer. 90% FPS and 10% what wait, I will go to this. What was the question? 90% FBS and 10% DMSO is correct. No, no. In the cell culture, usually 10% FBS, it is incorrect. Usually in case of cell culture, 10% FBS is used, and 5% DMSO is used. But we can use 10% DMSO also, but usually in cell culture 5% is used, because this DMSO is toxic to cells above 10%. So 5% is used, and this FBS is 10% FBS is used usually, but it depends on the condition. Sometimes 20% may be also used, but usually in most of the cases, if you say in a general way, so 10% is used. Any more questions? If you have any questions, you can ask here. I will try my best to answer. Or, or if I don't have the answer, I will answer. I may answer in the next session.
So for cryopreservant, you mentioned 5% DMSO
Yes, yes.
and 20% of FBS. So is it enough, or media also is added? No.
No, no. Media is also added, and in the media, this 20% and this 5%, it is maintained in the media also. Only media only. For example, you have the 100 ml of media, so you have to add 20 ml this FBS, that is a 20%, and 5 ml DMSO. But it
Okay.
though so you are talking about storing media.
Yeah, yeah, cryopreserve.
Yeah,
Uh,
cryopreservating media. So
can
you
can we preserve the cells only with FBS and DMSO without
huh,
media?
without media it can also be done. But as this FBS is very costly, you can also go with the 10% FBS. But if you are planning to see this store your cells for a very long time, you can go with 20% FBS. But if you have the resources and you are planning to store for a very long time, in that case only FBS will work. Only FBS plus DMS, but not required directly only FBS. You can also add the media and add 20% or 10% FBS.
Okay, okay. Thank you.
Any other question? So if you don't have any questions, so I can conclude this session then. So thank you so much for joining the session. You can go into my YouTube channel and you can watch if you have missed anything. And there are also some other videos present. You can also watch them. They may be helpful to you. So thank you for joining. Thank you so much. We will meet in the next session next Tuesday, coming Tuesday. Thank you.